Thank you. Hi, everyone. My name is Lendert. First of all, very grateful to be here and be able to present some of our new preliminary work in the group done by Nicole, Simon, and Nikita. And together with you, I would like to explore whether periodic temperature modulation is able to synchronize the cell division timing of early embryos. And we'll be talking about Xenopus levis frog. It's the African clawed frog, and it lives in these stagnant pools or slow streams. It's fully aquatic. It's also an ectotherm. So basically, all of its biochemistry is running at the temperature of the water that it's in. And here is its embryo. It divides every half an hour or so, quite periodically. And these embryos, they're in the same water as the frog itself. So it is developing over over hours to days. And in that period, it is in the water. So during the day, it's being heated by the sun, and then it cools off at night. So it's constantly being exposed to these varying temperatures during its development. And the cleavages, these early cleavages in this embryo that I've just shown you, they're really driven by a simple biochemical oscillator. And it's sort of beautifully simple because it can be explained by what I'm showing here. Basically, you have a protein called cyclin B, which is synthesized constantly over time from the maternal mRNA that is stocked in the frog egg. It then binds to CDK1, and when CDK1 is activated, it activates another protein complex called APC, which then leads to the degradation of these cyclins. So you've got this very basic negative feedback oscillator that drives these cell divisions. If we look at the oscillator, you've got like these periodic pulses where every time you've got a pulse in CDK1 activity, the cell divides. And as you can see here, it's pulse-like, so it's not like a sinusoidal oscillation, and that's because there are nonlinearities, there are differences in time scales, both of the synthesis, degradation processes, and activation, inactivation processes of CDK1. Now, what happens with the oscillator if the temperature changes? And that's shown here. What I'm showing you is the period of the cleavages versus the inverse of temperature. And the biochemical oscillator is driven by many different reactions that are interacting with each other. And every single reaction, in principle, is following the Arrhenius law. That is scales exponentially with temperature. So in principle, every single reaction is scaling linearly in this log versus inverse of temperature plot that I'm showing here. That's for a single reaction. And as you notice, the blue dots are the experimental measurements of cell division timing in function of temperature of this early embryo, and it is not following a straight line. It curves away from it, it slows down at the warm end, and it also curves away at the cold end. And so if you want to know more about this, I suggest you go to talk to Simon. He had a poster yesterday about this non-Arrhenius scaling in function of temperature, and I'm sure he's very happy to talk more about it during breaks. Shortly, what mechanistically can explain this non-Arrhenius scaling is the fact that you've got these two key processes, synthesis and degradation, and they've got a mismatch in how sensitive they are to temperature. So the scaling of these processes is different with respect to temperature. And that means that at cold temperatures, basically, the synthesis falls away faster than the degradation, so you get stuck in interphase at low CDK1 activity. And at high temperature, the synthesis speeds up faster than degradation, and so you get stuck in mitosis at high CDK1 activity. And at intermediate temperatures, you curve away from the linear response curve. So it's a network property that leads to this non-Arrhenius scaling because of a mismatch in these two processes and how they scale with temperature. Okay, now that we know this, I want to move on to fixed temperatures and look at the modulation of temperature. And in order to look at how the oscillator responds to changes in temperature, we built, together with a previous PhD student, Bartosz Prokop, this sort of system and this device which electrically controls the temperature on two sides, and it creates this spatial gradient in temperature over the device where you have a population of embryos that sits in it and feels all different temperatures at the same time, and we can image that live. And so what we do with that is you can get the previous plot that I've shown you in a single experiment. But now what we do is we start modulating this whole curve over time. So we modulate it up and down with a certain periodicity that from 20 to 70 minutes, for instance, on the same scale of the cleavage period, and with a certain amplitude up to, let's say, going 5 to 10 degrees up or down. Okay? And then we measure all of the cleavage timings of all of these embryos, and we analyze that. And we did this for various conditions, both for Xenopus laevis embryos, but also for zebrafish embryos, which I'm not going to talk about much. And so here are some initial findings that we had, the initial experiments, where plotting again the period of the cleavage versus the temperature, the base temperature with no modulation. And here in white, it's no modulation, it's just what I've shown you before. And then if you start modulating with a certain frequency, in this case here with the dashed line, you can see that the red ones are the cleavages now under modulation. They seem to sort of have a plateau here around when the modulation period is similar to the period of the oscillator. So we thought like, well, this looks like synchronization, which I was quite excited about. But then we took a step back and we first asked ourselves, okay, let's look at the oscillator and what do we expect an oscillator to do if you have this periodic driving of an oscillator? Rather than looking at the full complexity of the cell cycle oscillator, we started by looking at a simple phase oscillator. Basically, you move across a circle with a fixed phase velocity. So it's not realistic because the cell cycle oscillator is a relaxation oscillator. It moves at different speeds across the whole cell cycle oscillator. But we're just assuming that it's uniform right now. And then we took a look at two structurally different ways to apply this periodic driving. The first one is one we're all very familiar with. It's additive forcing, where we're adding this term, and you've got a driver here and the oscillator, and they're coupled in this Kuramoto-like way, where you've got the phase of the oscillator is coupled to the periodic signal of the driver. And so when they start oscillating, you've got a phase, they're coupled in phase, and they try to stay together as long as the frequency is similar enough to each other and the coupling amplitude is large enough. We can then also look at populations of these oscillators. So imagine this device that I've shown you. We've got all different temperatures. So all of these oscillators, they're dividing at different speeds, here shown in the different colors. And this is a simulation without any coupling, so they're just all doing their own thing. Now, when we introduce the coupling in this additive way, you can get this clustering and phase locking between all of these different oscillators. And if you increase the coupling, this clusters more tightly, for instance, and that's sort of the classic synchronization, Arnold tongues that you get, and so on, right? So the second way of modulating the system is parametrically, where I'm just taking the phase oscillator, and the period of the oscillator is now modulated periodically in time. So here in the animation, you've got, again, an oscillator that is moving around. And the color is showing you red. When it's warm, it's moving faster. When it's cold, it's moving more slowly. And so this is what you can get. We can, again, look at that for a whole population of oscillators. And in that case, they sort of move along the circle, and there is, again, diffusing. And there's no clear locking. Okay? So now we have these models. And let's look back at the experiment. So the type of plot that I'm showing here is every horizontal line is the cleavage timing for a single embryo in such a device that I've shown you. Okay? And they're ordered here from the bottom are the fast ones at the hot side to the cold side when they're slower. So they've got a certain period, and that's more or less constant over time. So the period at the different temperatures, we know what it's like. It's this non-Arrhenius curve. We know it from the experiments. We can plug it into the model, and the model shows something very similar. This is just no modulation, is the phase oscillator, how it behaves over time. Then we introduce the modulation. In this case, it's an experiment where we modulate every 35 minutes, here shown in the dashed line. And the other vertical lines in red and in green are showing when you go from cold to hot or hot to cold. So it shows you when you are in a warm phase or in the cold phase. And what you can see is that a lot of the cleavages, they sort of cluster together in the warm phase, while they are sort of absent in the cold phase. So it seems like the modulation is really gating the cell division to occur in the warm phase, and especially for the periods that are slower than the modulation, basically they don't manage to divide in the hot phase, so they decide to skip during the cold phase and divide in the next one. So you have these phase slips, basically. So this is what we saw in the experiment. And here's the same theory based on the phase oscillators, where you have these same staircases and also the phase slips that you see for periods at the colder end that are slower than the modulation period. And so this is all the time I have for the experiments. It's just one example, but it seems like this works pretty well. We can also look at it in a different way. So here I'm showing the cleavage period in function of the base temperature, again in white without modulation. And then with modulation, you can see that there are two things that happen. There are two branches. In red here, you can see that, well, they're not on top of the line, which would be phase locking, but instead they're sort of pulled towards the red line of modulation. But then there's also this second branch over here, which are the phase slips, and they divide much more slowly. Okay? And again, here's the theory, which sort of shows the same type of thing. And I think that's sort of I'm out of time. So I started with the question whether synchronizing embryonic cell cycles by temperature modulation is possible. And so the answer is not quite. And there are two messages. That is that the non-arrhenius scaling is a network property due to the mismatch between scaling of synthesis and degradation processes. And the temperature acts as a parametric frequency modulation and is gating the cell cycle to divide in the warm phase. You also have these phase slips, and there's no phase locking. And with that, I would just like to thank everybody in the lab, especially Nicole, who did all of the experiments and the painstaking analysis of all of the movies, and Seaman and Nikita, who did the analysis. And so Seaman is also here with a poster. So feel free to come and talk to either of us. Thank you very much.